Hello, and welcome to my Claymation video on the topic of alternative splicing. Before we learn about alternative splicing, let's first talk about the normal splicing process. To make proteins for the cell, we know that the first step is for DNA to be transcribed into mRNA. This mRNA must be processed so it can be considered mature. To do this, introns in the mRNA, shown here in red, must be spliced out. This is done by the spliceosome. Once the introns are removed, the exons are joined together to form a continuous, mature mRNA. This is the normal splicing process. In the three different kinds of alternative splicing, things happen a little differently. Different combinations of exons are included in different splice sequences. The first kind of alternative splicing we will discuss is called cassette exon splicing. In cassette exon splicing, a certain exon may or may not be included in the final RNA product. As shown, one form includes the exon while the other form does not include the exon. These two RNA products will result in different products once translated. For example, including the exon may result in precursor chondrocytes found in cartilage, while splicing out the exon may result in mature chondrocytes. The second kind of alternative splicing we will discuss is called mutually exclusive splicing. In mutually exclusive splicing, there are at least two exons that will not be included together in the final RNA product. Either one or the other will be spliced out to leave just one of them. As shown, these two exons are mutually exclusive, and splicing one versus the other will yield different final products. For example, if the left mutually exclusive exon is spliced from the RNA, this RNA may code for limb ectoderm. But if the right mutually exclusive exon is spliced from the RNA, this RNA may code for limb mesoderm. The third kind of alternative splicing we will discuss is called the alternative splice site. As we mentioned earlier, normal splicing involves splicing all of the introns out of an RNA. But with an alternative splice site, the spliceosome can cut at different locations besides the ends of the introns. Alternative splice sites are located on exons, and it allows the spliceosome to cut the intron as well as part of the exon out during splicing. As you might guess, including different amounts or regions of certain exons will result in different final products. There are two main categories of alternative splice sites: alternate 5' and alternate 3' sites. An alternate 5' splice site is just what it sounds like. It means the spliceosome cuts at a normal spot on the 3' end, shown here on the right side. But on the 5' end, the left side, it can cut at the alternative site located on the exon. An alternate 3' splice site is also just what it sounds like. The spliceosome cuts at a normal spot on the 5' left end, but on the 3' side, the right, it can cut at the alternative site, which is located on the exon. With these different types of alternative splicing, the same transcribed mRNAs can be processed in many different ways to make many different products. Alternative splicing allows the cells to use the same DNA to code for many protein products, contributing to the great diversity that we see within our cells. I hope you enjoyed this video. Thanks for watching and have a great day!